Morning, internet. Ugh, and I just woke up. This is a daily stream where I try to understand the latest scientific literature by browsing the figures published in top scientific journals. My name is Miles, and I'm a biologist slash chemist slash mathematician slash curious person slash biology nerd, and I will be your guide on this journey. I hope you are buckled in because we got a pretty interesting paper to start us off here. It's about how the nutritional quality and insect resistance of plants declines under elevated CO2 levels. Um, I assume that refers to uh, elevated CO2 levels as experienced during climate change. And yeah, let's dive right in. Go ahead and start with figure one. All right, gotta love a good old bar plot. Nothing like a bar plot to start us off. Simple, intuitive. Let's try to understand here. So here we have measures of plant biomass, the mass of tissues. Looks like the top bars look at shoot mass and the bottom bars are root mass. That's interesting. I've never seen a bar plot like this with bars reflected across the x-axis. I don't know. It's kind of strange. I would think that it seems like it'd be easier to compare these if this roots bar wasn't reflected across the x-axis. They were just side by side. It's not like the y-axes are different scales. Anyway, so it's like white bars and gray bars. I assume those are different levels of CO2. Okay, yes, so I think normal CO2 levels are the white bars and elevated CO2 levels are the gray bars. So increasing the amount of CO2 exposed to the plant increases shoot mass and increases root mass, it looks like. Or it looks like the difference in root mass isn't significant. I guess this increase in shoot mass makes sense since plants their elevated CO2 levels, plants can fix more carbon and build up more plant tissue structures. Foliar nitrogen concentrations. Let's see, so concentration of nitrogen in foliage, I guess, comparing normal CO2 versus elevated CO2. It's an interesting decrease when CO2 is increased, or decrease in nitrogen concentrations um, when carbon dioxide concentrations are increased. Marginally significant. Interesting. Oh, looks like these guys are also investigating root modulation. Let's see here. So I think in this paper, so scientists were investigating a plant called Medicago sativa, and that is a very common model legume in scientific research. Legumes are typically what you think of as beans and peanuts. You can get a nice picture of here. Same as alfalfa. And the interesting thing about legumes or plants in the same family as alfalfa, they have a what's called a symbiotic relationship with certain kinds of bacteria that live in the soil. Soil bacteria will invade the roots of leguminous plants, where the plants will house the bacteria in little outgrowths called nodules. And in those little nodules, the bacteria will fix nitrogen for the plant. Let's see if we can get a picture of nodules. Nodules on fingers. Why is that the first thing that comes up? Gross. Nodules on legumes. Just go into Google and Google nodules on fingers. So these are kind of what nodules look like at plant roots, and then these uh, little, or sometimes very large bumps on the roots. That's where the bacteria are housed and where they uh, take nitrogen out of the atmosphere and convert it into a form that plants can use for growth. So it pertains to this first figure where um, it looks like when concentrations of CO2 were increased, um, these plants formed more nodules. It's pretty interesting. I wonder why that would be. Perhaps um, I don't know. Let's see here. And here we have carbon to nitrogen ratio in foliage. Doesn't really change all that much. These guys really only have marginally significant results, which is surprising. All right, on to figure number 2. Let's see, these guys have 4 figures. I don't know if I'll look at all of them. Look at least a couple. Let's look at this one next. Okay, so next they examined concentrations of amino acids in foliage tissues. Amino acids are building blocks of proteins, and they compared amino acid concentrations under ambient versus elevated CO2 levels. Looks like total amino acid concentrations don't change that much, but concentrations of specific amino acids decrease under elevated CO2 levels. It's like leucine and arginine levels decrease, as well as tyrosine. You can barely see those little baby bars. Okay, that's interesting. So that might be where they claim that the nutritional quality of plants declines under elevated CO2 levels. That's pretty straightforward. This is what I love bar plots because they're always just so easy to interpret. So easy to understand just at a glance and uh, relatively straightforward to understand. Okay, so now these figures get into herbivore resistance. So JA, that probably stands for jasmonic acid. It doesn't say here, but it's probably jasmonic acid. Jasmonic acid is a plant hormone that is involved in plant responses to stress and tolerance to various abiotic stressors. So looks like under elevated CO2 levels, plants produce more jasmonic acid, which is interesting. Did I mix up the white bars and gray bars? Oh, so now the white bars and gray bars mean something different? Okay, so the gray bars are represent plants that have insects uh, attacking them, and then the white bars are plants that don't have insects attacking them. And it's like these two different sets of white bars represent ambient and elevated levels of CO2, respectively. It's not entirely clear. Oh, here it is down here on the x-axis. Yeah, so the left bars are ambient CO2 levels and are elevated CO2 levels. Um, so yeah, it looks like you add insects to a plant, the plant starts producing jasmonic acid. That's not it's not too crazy, but it looks like under elevated CO2 levels, plants don't produce, or the jasmonic acid response that's normally induced is much weaker, which is interesting. So I guess since jasmonic acid is often involved in um, responses to herbivory, perhaps these um, decreased levels of jasmonic acid could be making plants more susceptible to herbivory. It would have been nice if they measured herbivory or some measure of plant damage and plotted that. That would make for a more direct comparison to their claim that um, insect herbivores have increased performance under elevated CO2. But we'll see. And this is interesting. So RGR this probably stands for the rhizobial growth response. In other words, or maybe not. Let's see here. I'm not familiar with these units. What I thought this was getting at is comparing growth of plants that have nodulating bacteria and plants that are not given nodulating bacteria, and see that plants get a larger boost to their growth from these nodulating bacteria under elevated CO2 conditions, which is very interesting. It doesn't make much sense if the nitrogen amino acid content of the plant is also going down. You'd think that if uh, if um, these soil bacteria, these nitrogen fixing bacteria, are boosting the growth of plants more, perhaps they're fixing more nitrogen for the plant, then you wouldn't expect the nitrogen content of the plant to also go down. Kind of counterintuitive based on their previous figure. Let's see if I can bring it up. You might not be able to see it here, but yeah, they show that nitrogen concentrations in the plant decrease a little bit, as well as the concentrations of specific amino acids, yet um, these nitrogen fixing bacteria are boosting the growth of plants a lot under elevated CO2 conditions. It would have been nice if they measured nitrogen fixation rates or something like that to try to explain that discrepancy. It's kind of strange. And oh, it looks like this is the last figure, so look at this figure and then we'll go on to another one. I'm on the website for, yeah, this is Nature Scientific Reports, which is a pretty well-regarded sub-journal of nature, I guess you could call it. So it looks like these guys are looking at effects across a bunch of different scientific studies in what's called a meta-analysis. It's kind of cool they did that. Let's see. Okay, so they looked at a bunch of different studies showing how elevated CO2 levels affect the performance of this one herbivore. What is this herbivore? I've never heard of it. Armigera? Sounds like Armageddon almost. Go ahead and close these. Let's get a picture of this guy. Oh, it's just a cute little caterpillar. Cotton ballworm. Ballworm? Cotton ballworm? Oh, that's cute. Looks kind of pretty. A huge agricultural pest, though. A nice caterpillar, too. Oh man, looks like these guys eat everything. Cotton, tomatoes, maize, chickpeas, alfalfa, and tobacco. Okay, so the points here are divided up amongst a bunch of different plant families, it looks like. So Solanaceae, that's the family of plants that includes like tomatoes, potatoes, eggplants, peppers. Pretty awesome family. Poaceae, that's the family plant that that's a plant family that includes lots of grasses like wheat, corn, um, rice as well. I'm pretty sure. Um, Fabaceae, that's the plant family that includes like common beans, peanuts, lentils, split peas, and so on. Malvaceae, I've never heard of. What's Malvaceae? Malvaceae. It's probably a family that has a lot of crops in it. Scientists really only ever get paid to, or at least plant biologists, mostly get paid to study crops. So most research in plant biology is done on crops. These look pretty though. Mallows. Economic importance. Okay. Produce okra, cotton, and cacao. And durian. Ugh. Durian? I think durian's like, it's an infamously, yeah, it's one of these guys. I think it's an infamously awful smelling fruit. It's supposed to smell like sweaty socks or something, yet people love it. So anyway, let's see here. So yeah, they're looking at how elevated CO2 affects the um, herbivory of this one caterpillar across a bunch of different studies. Looks like a lot of the studies found um, decreased herbivore performance under elevated CO2. So it looks like, since these points lie on this line beneath zero, though a couple of them have error bars that overlap zero, so can't confidently say whether or not performance of this herbivore is increased or decreased under elevated CO2, which is interesting. Yeah, points to the left indicate significant decreases in performance. Does that conflict with what they showed in the previous figure? I think they showed increased performance, didn't they? I think they did. Or I don't think maybe they didn't measure performance. They only measured, or they only reported measurements of jasmonic acid concentration, which I don't know if that's really a measure of the herbivores' performance. You want like a more direct measure of herbivory, like, I don't know, like amount of plant damaged or some measure like that. You might have reported something like that in the supplemental, but I think I'm going to go on to a new paper. That was a good paper. I love how simple and straightforward the figures were. Figures I've been looking at recently, some of the previous streams, like, oh man, I was getting a little stumped on every panel. Let's see. I'm gonna check out the genetics section again. Let's see, where am I? Okay, here we are. Typically go browse the genetics section just because genetics is my favorite science, but if anyone ever joins me, they're welcome to drop suggestions of papers to look at in chat or anything like that. I don't really care too much what that. A lot of these, all these papers are pretty interesting. So let's see here. Try to find a recent one. C. elegans, rice. Never heard of that. Comparative genome analysis. Splice genes. Muscle from some like producing meat. Bluefin tuna. Mycobacteriophage. Toxic genes. Let's see, how am I doing on time? Okay, in about 20 minutes, so probably look at a couple more figures and call it a day. Oh man, button mushroom genomes? That sounds cute. European cattle again, dietary intake, phenylketonuria. Oh man, study about chickens? Chinese gamecock chicken? Oh man. Chinese indigenous chickens. Alright, let's check this out. This sounds cool. Oh man, figures are very colorful and pretty too. I feel like this will be a good one. Alright, my world geography is terrible, but this looks like a map of China, so not lost yet. Let's see here. Here they're showing genetic diversity across, it's probably a bunch of different genotypes of chicken, I'm guessing. Here they're showing heterozygosity across the different chicken types. So heterozygosity is where state is a state where the genome copies an organism gets from their mother and the copy they get from their father. They have different structures at a given location. And that state where the two copies differ at a given location is called heterozygosity. They're said to be heterozygous at a given location. And then here, linkage disequilibrium. How to explain that? So linkage disequilibrium is a state where two regions of DNA tend to occur together in the same genome. Yeah, maybe I can get a picture of it. They occur together more often than you would expect by chance. No, it doesn't look like this has a picture. A non-random association of alleles, which are just variants of a given DNA region. Um, yeah, so non-random association of DNA variants. It looks like these associations decay as something increases. I don't know what the D genome heterozygosity 22 popular. Wait, they mixed up their they mixed up um, their captions for figures C and D. C refers to linkage disequilibrium. Sorry, no, panel D is the linkage disequilibrium figure, and panel C is the heterozygosity figure, but they mixed up the two right here. Surprising they didn't catch that before publishing it. See here. I'm not sure what's on the x-axis right here. Like, what is increasing? I don't know. This is why you always label your axes. It's kind of funny how sometimes on figures for top scientific journals, they won't follow like the most basic advice that you get for making figures as an undergrad, like always label your axes, always start your y-axis at 0, and things like that. But it's kind of funny how these incredibly advanced, smart, uh, intelligent people, they won't catch things like that. That's not always a huge deal, but it does make understanding the figures just slightly harder. And so now they're showing relatedness among a bunch of different genotypes of chicken, but a couple different clusters of chicken types: chicken group 1, chicken group 2, chicken group 3. We need a picture of a gamecock chicken. Let's see here, gamecock chicken. I assume it's from like, these are the kinds of chickens that people have fight one another. Yeah, yeah, exactly. And they're very pretty. This one's like chained to a pallet. Let's see here. Now they did a principal component analysis to this is just another way to show these different clusters that they found in their data set. Let's see, what's this here? An admixture analysis. I'm not sure what an admixture analysis is, but whatever it is, they're showing how it varies amongst a bunch of different chicken types. You can see that the commercial lines, those are all one color, whereas these wild chickens, I guess, in a sense, or non-commercialized chickens have a bunch of different colors. Perhaps that refers to like genetic diversity of some kind. Let's see, what is an admixture analysis? How am I doing for time? Okay, probably look at just one more figure on chickens and then good. I haven't quite figured out how to like start these streams. Like, feel like I should have some kind of fancy intro or something like that. Maybe a theme song. That's in the works. I might just bring out my little tiny piano and smash the keys on that at the start of every stream. That'll really raise the production value. genetic admixture, presence of DNA in the individual distantly related population or species. Okay, so genetic admixture refers to an individual having DNA from a distantly related species as a result of interbreeding. Okay, so I guess there's more interbreeding among all these different gamecocks versus the commercial lines, which makes sense. I think I'll look at one more figure. Come on. There we go. I'll call it good. Man, these guys have 6 figures on chickens. Or 7 figures on chickens. Man. This one looks kind of crazy. Showing migration between a bunch of different chicken genotypes, perhaps. Let's try to look at look at this one. This one looks cool. So ZST versus ZHP. They're looking at how some quantity varies across chicken chromosomes. And chickens have 27 chromosomes. I was not aware of that. Select signatures. Okay, so I guess they're showing what regions of the DNA have been selected for during the breeding of these gamecock chickens. It's interesting, they have a lot of hits. There are have a lot of genes labeled here. Looks like the same genes were picked up, whether they calculate selection using the ZHP or ZFST statistic. It's pretty good correspondence between them. There are only like a couple that are present in the ZFST and not present in ZHP. That's cool. I wonder what oh, this BDNF is an important candidate gene concerning gamecock behavior. Oh man, so oh man, but BDNF didn't pass their threshold. That's interesting. So this gene is known to play a role in gamecock behavior, yet didn't pass their selection threshold. Interesting. Those genes are involved in. Well, I've been going for about 30 minutes, so it's probably about time I go try or I stop recording and get back to my other work. But yeah, if you joined me today, thank you for spending a little bit with me, and I hope you have a good rest of your day. I'm not sure. I'm still working out whether or not I'm going to stream daily or twice a week. I'll probably end up streaming twice a week eventually, but for now I'm trying to do it on a more daily basis as I iron out, as I experiment with different ways of setting up the stream and the different ideas that I have. So more to come on that soon. Anyway, have a good rest of your day.